So we'll just give it a fefew more moments and then I'll explain how it's going to work. In the meantime, a very, very warm welcome. Jasmine and I have met before this and we were both very excited about doing this session. Over the last year, we haven't had many opportunities to engage with undergraduates, and we always really enjoy these sessions. It's the second year in a row we've done these, and we're very much looking forward to welcoming you all here online to UCL. So, so we're on 1 o'clock. So I'll just explain how the system is going to work. So if you have any technical problems at all, please put in the chat if you can't hear us clearly or there's any kind of problem with the video, then please let us know and we'll try and solve your problem that way. You can ask questions throughout the session at any point. There's a Q&A box. You can put a question there. We may wait until the end of the section that we're in to try and bunch questions up together. Or, you know, we've got thematic questions that seem to be asking similar type of things. Or if somebody's asking a question that's easier for me to answer in text and put a link in there, I may do it that way. But you can ask a question at any point and you can ask it anonymously. We've got 2 polls that we're going to launch. We'd just like to know whether you're interested in undergraduate or postgraduate study or neither, and whether you have an offer to study with us, just so we've got an idea of our audience from this point. And at the very end of the session, we'll send you a link to a webinar. So what I'm going to do now is I'm going to hand over to Professor Martina Micheletti, and after that, we're going to have Jasmine. And as I say, ask questions throughout, but we may wait until the end of that section before we answer them. If I turn my camera off, I am in the background, so I'm here answering your questions. Over to you, Martina.
Right, so can you see my slides correctly?
Yeah, great.
Okay, perfect. So my name is Martina. I'm one of the professors in the Department of Biochemical Engineering. And today, yeah, it's a pleasure to be here. What I'm going to do is just to give you a brief introduction to a little bit UCL department leading to how I work in the vaccine manufacturing space. And then, yeah, introduce one of my researchers, Dr. Jasmine Samaras, who is going to, you know, provide an example of her exciting work. But first, something about me. So you know who you're going to talk to, who you're talking to now, and who you're asking questions to. I'm from Italy, as you probably understand from my accent. I have done my studies there. I always had an interest in the bio side of things, but in Italy it wasn't available as a first degree. But I did have the opportunity within that to visit and well, to spend a few months in Birmingham as part of my degree, and there I was working as well for a company for a period, working on some bacterial work in bioreactors, which was you know, which really fascinated me. I then was offered a PhD, so I did a PhD in mechanical engineering. So you see how my background is really sort of multidisciplinary already, although really focusing on the engineering fundamentals at King's College. And it was a great experience where I realized that research was for me. And again, there I was, you know, again, focusing a little bit more on what we call upstream processing, and specifically reactor works and reactor fundamentals. So there was a position at UCL. So I was at UCL as a postdoctoral researcher, and I've been here since 2004. First, I was working in interdisciplinary project with different biochemists, biologists, and chemists, which was amazing. And then I joined as an academic staff member in 2007, and I became a professor a couple of years ago. My interests now really focus on engineering fundamentals. So in the department, we have different staff with very different expertise. So those of you who attended Duigu's lecture, she is, you know, as well an engineer focusing on really the modeling side. I'm an experimentalist, and the rest of the department is really, again, comprises, staff with expertise in the life sciences as well. So quite a mixture. So as I was saying, very focused on the engineering and especially the impact on the environment, on different cells and the product. I have applied the portfolio of techniques that I have developed for, looking at the hydrodynamic environment to different types of products in both the cell and gene therapy space, to biopharmaceuticals, and more recently to vaccines. In the department, each academic staff member has different roles. So for those of you who will join us next year, I will also be your departmental tutor. So essentially, I'm responsible for pastoral care in the department, but also have this role of overseeing a number of different matters and your experience at university from the time you join, after admissions, during the teaching, assessment, and communicating with the students most effectively. So you might have researched about UCL, so I'm not going to spend too much time focusing on these. But those, for those who joins this seminar for, as one of the first. So we are in the Faculty of Engineering Science. Is very successful for grant funding recipients. What does it mean? It means, we do lots of research that actually has an impact, and we are successful with that. In the department, first department of its kind, and we are again focusing a lot on different applications and cutting edge applications, and have quite a strong research and teaching base here. So something more about the department. We consider ourselves as one of the largest academic bioprocess group in the world, who's focusing mainly on really bioprocessing, rather than proposing this only as a side sort of research topic. We are indeed a small department. They're much bigger engineering department. So our intake, for example, at undergraduate level is around 40, 50 students. But we are very happy with the type of staff to students ratio that we propose. Large cohort of doctoral researcher and postdoctoral, and I think we are also very proud to to be really like family and very friendly environment. Definitely multidisciplinary and a team-based approach. We have a lot of academic and company collaborations. So we have a number of different hubs, and I'll talk about one of these in a second to introduce the vaccine space, which students really benefit from, as everything we do really expands into our DOT programs and research projects as well for undergraduate and postgraduate DOT programs. So what type of research do we do, and what type of products do we do research on? So approximately the department is divided into 3 different areas: the industrial biotechnologies, which works essentially when working on complex chemicals, better pharmaceuticals, but also finding environmentally friendly solution. We work on biopharmaceuticals, so different types of therapeutic proteins, gene therapy, working for different viruses, and vaccines belongs to this category. And then cell and gene therapies that you might have heard from. So really the stem cell work and gene therapy work as well. And some of us have got expertise that actually goes, spans across all these different area. One of these areas is vaccine, which we're going to talk about today. And it's something that I embraced recently, as I'm the director of one of vaccine manufacturing research hub. What's a hub? It's really like a group of researchers. So we have this sort of large grant which funds researcher, and it's a very difficult environment to be in. The landscape for vaccine manufacturing is really limited investments. And when I say traditional here, I mean before the pandemic, as a lot of things have changed after. Limited investments. It's really the process development for vaccines is complex and therefore costly, high risk of failures. Many times for a vaccine against a specific pathogen, we have to develop a new process. As I say, manufacturing is complex, and of course there are, in association and alliances like Gavi negotiate United, within the United Nations, they essentially negotiate quite a low price for per dose. So really we need to bring the cost down to the cost of production. So within this context, a few years ago, me and Sarah, that you might have, Professor Sarah Gilbert from the Jenner Institute in Oxford, that you might have heard in the news as one of the leader of the Oxford AstraZeneca vaccine. We essentially are the co-directors of this VaxHub, and we have worked with different companies, different low and middle income companies, members, and as well as other partners on research. So what do? We look at developing, scaling and manufacturing vaccines for the benefit of low and middle income countries. So this is to ensure that we have supply of these essential vaccines. We work with manufacturers, for example, from Indonesia, from India, from Vietnam, to ensure that these advances are translated to, the technologies are translated to manufacturers so they can do their own production. And we propose technologies that are able to support an epidemic threat. So because, and as I say, this is a proposal that we did prior to 2018, so well before the pandemic. And it is really, can we have responsive technologies that are able to respond to different type of epidemic scenarios? And of course, some of these technologies then turned out to be quite good and beneficial, to prepare and to produce a vaccine against the current pandemic. So this is our team, UCL and Oxford, but we have other academic partners in different university, University of Leeds, Imperial, and London School for Hygiene and Tropical Medicine. So this is about some of the research in the hub that, as I said, permeates throughout the taught degrees. We build on the fundamental first, and then we have full courses in the final year for, on vaccine bioprocessing, which is, I guess, the most relevant to this topic. And on this note, I'm going to stop sharing here and introduce one of my researchers. This is Jasmine Samaras. So if she can share her slide. And she was in our degree, and she is in my group. So she's been with me for a PhD and a postdoc. And she's going to, and now works in the VaxHub with the London School for Hygiene and Tropical Medicine. And she's going to explain a little bit more about what she does and what's the really the excitement of the of a new technology that we collaborate with the London School.
Sorry to interrupt. Just before you start, Jasmine, just to say we've had one question come through. We'll keep that until the end because it's a really, really good question. But any more, please bring them in. I'm also just about to launch a poll, so if you just fill that in as we go along, it'll be really interesting. Thank you. And sorry to interrupt. I'll let you carry on.
Okay.
Okay, so thank you, Martina, for your introduction. Hello and good afternoon, everyone that's joined us today. Thank you for taking your time and joining us. I'm going to be giving your taster lecture on vaccine manufacture. I hope you find it informative, interesting, and maybe see something that you like and want to take forward in your careers. So my name is Jasmine Samaras. So I got my master's and my PhD both in the Department of Biochemical Engineering here at UCL, working with Martina. My PhD was sort of looking at combining the field of engineering with stem cell research. So we were looking to improve upon the current yields for stem cell repair, specifically looking at repairing the heart. And that was a collaboration with IBET, which is a research laboratory in Portugal. So I actually got to go over to Portugal for my PhD, which was really cool. I'm now currently a research fellow in the department. So I'm working on vaccine research, as Martina said, and using engineering kind of know-how to improve and try and help vaccine manufacture processes. So I guess this kind of takes me on to the end of my presentation, where I'll give you a little bit of an overview of the work that I'm doing and how that relates in general to kind of undergrads and if you come to join the department. So, why is that not changing slides now? Okay, so vaccines are really vogue at the moment. They're gaining so much attention all around the world with the outbreak that's happening around us. But what is often forgotten is the historic origins of vaccination. So where did the word come from? How was it discovered? So for this, we remember Edward Jenner, which coincidentally is where the Oxford team responsible for the now AstraZeneca COVID vaccine got their namesake. So in the 18th century, we were plagued with a virus known as smallpox, which caused approximately 400,000 deaths a year, which doesn't sound like as much as what we're seeing at the moment, but in those times there was a much smaller population in the world, and it was quite a significant number of people, and a third of all survivors were going blind. So it's not a very nice disease. Jenner observed that milkmaids in his local area had been previously infected with much milder cowpox, and then appeared to be protected from smallpox. So he kind of experimented a little bit with this idea and found a milkmaid, her name was Sarah Nelms, who had fresh cowpox lesions. Apologies for the image, it's kind of gross, but just to give you an idea of how it looked. He used a sample of her wounds to deliberately infect an 8-year-old boy called James Phipps. He then recovered from cowpox, which he was indeed infected with, thanks to Jenner. But then when he tried to infect the boy with smallpox, he found that he was protected. So the Latin word for cowpox is vaccinia. So Jenner decided to call his new procedure vaccination. And thanks to this discovery, naturally occurring smallpox has now been globally eradicated. So thank you, Jenner. So what is a vaccine? By definition, a vaccine can be any component or preparation which can be used to stimulate an immune response. These components that you kind of prepare are called antigens. So vaccines create immunity by introducing either a weakened, inactive, or a subunit of a pathogen that makes us ill, such as bacteria or viruses. The acquired immunity from a vaccine essentially then prepares your body to mount a defense when it encounters the real disease-causing agent. So before COVID, the World Health Organization, or the WHO, roughly estimated that there were between, or at least 2 to 3 million people saved each year through vaccination programs. So vaccine preventable diseases include things such as rabies, flu, tetanus, cholera, hepatitis B, bacterial pneumonia, measles, and of course COVID-19 can also be prevented with vaccination. Disease variants are then another matter and may require, for example, a new vaccine, which is what we see, for example, with the seasonal flu jab that every year is rolled out, at least in the UK. I can't speak for all the other countries that have joined us today. So thinking about the typical vaccine timeline, a vaccine from initial discovery in a lab can take anywhere up to 15 years before it is then approved and reaches the market. So this encompasses the initial research phase, when we are dealing with thousands of different possible vaccine candidates, to then progressing through to preclinical and clinical phases, where fewer and fewer vaccine candidates are screened for things like safety, efficacy, long-term impact, and dosage. As the number of vaccine candidates decrease, the number of participants in the clinical trials increase, but then coincidentally so does the cost. If we consider the overall cost, you're looking at anything between 500 million and up to $5 billion per vaccine, which is just an incomprehensible amount of money. And it varies so much with manufacture, the vaccine itself that's being developed, the country that it's developed in, the facilities, do we need to contract out manufacture? There really is quite a lot to consider. So during an epidemic or pandemic scenario, these timelines are hugely reduced, as we have seen in roughly 12 months from discovering the virus. The Oxford AstraZeneca jab, for example, was approved in the EU in 12 months. When you're looking at this kind of normal timeline of 15 years, that is super impressive. So a vaccine can take one of many different forms. The design is predominantly dictated by the nature of the pathogen and how it affects us, as well as practical considerations, such as how the vaccine will be used. Most common are live attenuated vaccines, where the infectious agent is altered to be less harmful and in a kind of weakened state. Inactivated vaccines are another of the most common, where the infectious agent has been killed. And then there's also subunit vaccines, which essentially just inject a portion of the infectious agent. So generally speaking, subunit vaccines are considered to be the safest and also the most efficient of the three. I also thought it'd be interesting to highlight the three currently approved COVID vaccines in the UK. So the Oxford AstraZeneca vaccine is a viral vector-based platform where essentially genetic instructions of the coronavirus are transported into the cell via a weakened virus delivery system. The Pfizer and Moderna vaccines are mRNA vaccines, which again provide genetic instructions to the cells, just without the viral delivery system that the Oxford AstraZeneca jab uses. So in the context of my research, I'm going to focus on conjugate vaccines, which are essentially a type of subunit vaccine. Sometimes just using a subunit of an infectious agent in a vaccine is not enough to get a sufficient or long-lasting immune response. So to improve this, we can combine this weak component or a weak antigen with a stronger immune boosting antigen and create what is called a conjugate vaccine. So a good example of a conjugate vaccine are vaccines targeted against pneumonia, which is caused by the bacterium Streptococcus pneumoniae. So there are 90 different forms of this bacteria that cause pneumonia, which are called serotypes. So luckily for us, only a few of these serotypes actually account for most of pneumococcal disease. We don't see all 90 affecting people to the same extent. So the diagram on your left shows the bacterium Streptococcus pneumoniae with some of its kind of main features highlighted. You'll notice on the outer layer, there are these kind of branched, fluffy looking chains. These are called polysaccharides, which are essentially just made up of sugar molecules. So vaccines designed against pneumonia target specifically this cell surface polysaccharide, which is specific to each of these 90 different serotypes of a pneumococcal disease. So the vaccines against pneumonia are either made up of just this polysaccharide unit, which is essentially partially protective in adults and not for as long a duration, or they can be coupled, or this word conjugated, to a kind of protein toxoid component, which essentially just makes a more potent vaccine and makes it much more effective in the elderly as well as children who have more kind of naive immune systems. So these pneumonia vaccines are a lucrative business. So in 2019, Pneumovax 23, which is manufactured by Merck, made the company $334 million. So this is a polysaccharide only vaccine, so is best suited for adults and doesn't necessarily offer good long-term protection when used by itself, But as the number at the end of its name kind of suggests, it covers 23 of the 90 different serotypes of pneumococcal disease. Prevnar 13, which is manufactured by Pfizer, is a conjugate vaccine. So it's made up of the polysaccharide component conjugated to this immune boosting protein component. Now, this made a huge $5.95 billion in 2019, which is serious money. But pneumococcal disease is still a pretty big problem globally. In 2017, for example, pneumonia was responsible for the deaths of 2.56 million people. A third of these were below 5 years of age. So despite there being these vaccines available on the market, they're not reaching all of those that need them most. And this is largely related to the cost of these vaccines. So the reason that they're so expensive is mainly down to the production process that's necessary to manufacture them. So I'm going to focus a bit on Prevnar 13, for example. So this is made up of 13 different serotypes, which means that in manufacture, we need 13 separate cultivations to produce the polysaccharide for each serotype. Each of these must then go a purification process, which, although simplified on the left, is actually quite a complex and multi-step process. So on the right, you can see the kind of all the steps. So in our degree programs, you'll study each of these individual unit operations, learning what they're for, when to use them, and even design your own manufacturing process for different applications, maybe one like this. So each of these individual steps, so you've got centrifugation, filtration, chromatography, they all cost money to perform, and each of these steps will result in a loss of your yield. Now, a loss of yield means a higher cost at the end because you're getting less out of it from what you start with. In addition to this, so in addition to these 13 steps already that need to go through all of these steps in between, we must also separately cultivate this immune boosting protein component, which again needs its own chain of purification steps before it can then be combined or conjugated with this polysaccharide component. So once you've combined the polysaccharide with the protein, you then need to go again with another purification chain to finally get this product to meet the requirements to be a vaccine. So imagine doing 3 huge purification chains 13 times, and you can kind of begin to understand why this process is so expensive. So the complexity of it is is only made worse when you factor in quite often these steps are not all done in the same facility. They're done by lots of different scientists. So you also have to account for things like storage, transport. Also, you're maintaining ultra sterile conditions here because this is, at the end of the day, a vaccine that is going to be injected into healthy people, and it is just expected. So this, why can't countries afford this vaccine? Because Prevnar 13 can cost anything up to $169 per dose. That's huge. So luckily, and Martina kind of mentioned these guys in her talk as well, there are global alliances such as the Global Alliance for Vaccines and Immunization, or Gavi, which actually aim to subsidize and make vaccines such as Prevnar 13 more accessible and affordable for low-income countries. The map on the right shows you the countries that are eligible for Gavi support. Those that can and do benefit from Gavi can access Prevnar with reduced pricing, with anything as low as $3 per dose, which is great. But there are still quite a few lower and middle income countries that lose support of Gavi who still can't afford this vaccine, which is countries such as Angola, Bolivia, Indonesia, Sri Lanka, just a few, for example. So we clearly need innovation in this space. We need something new. And that is some of the work that myself and my colleagues at UCL hope to contribute to. Is it going to change? Okay, there we go. Sorry. So a really promising alternative to the current manufacturing process is a bioconjugation method where the conjugate vaccines are actually produced and kind of conjugated or coupled inside the cell of E. coli. So you've all heard of E. coli, perhaps more for the wrong reasons than the right reasons, but in the world of pharmaceutical manufacture or just in general kind of more biological world, E. coli can be engineered pretty easily to be a powerful workhorse. And in this context, it can offer a significantly simpler production process for conjugate vaccines. Oh, sorry, I think I leant on my mouse, there we go. And in doing so, it can offer a really good alternative to conjugate vaccines and hugely reduce the cost. And let me show you why. So if we envisage the production process for these bioconjugate vaccines, where they're expressed in E. coli, first, the actual cultivation and combination or conjugation of the polysaccharide and protein components are all done in a single step inside the E. coli cells. So all we have to do is cultivate and grow E. coli. Then, instead of these 3 separate purification chains that we were seeing before to get our product, we only need 1 purification process to go from this initial cultivation of E. coli to our vaccine. So less process steps is basically less material cost, quicker production, and a significantly, less losses in your yield. So hopefully this should give us a much cheaper product. So there are some products that are made in this way, using this technology, this bioconjugation technology, which are in clinical trials. But this is still a relatively new technology, still a new idea. And there are no vaccines commercially available that are made in this way. So how do we speed this up and how do we get this to market? Because this could be fantastic for the world to have. So coming back to our vaccine development timeline, myself and my colleagues are currently in the early research phase with our pneumococcal bioconjugate vaccine. So this means that we are still optimizing production as well as the product, and therefore have thousands of different combinations and vaccines to test. Yes. So in the department, we use a number of different tools for bioprocess development. The first example is miniaturization. So when we cultivate E. coli to produce our conjugate vaccine, this can be done in a number of different ways in different volumes. So the photos that I've got up on the slide are just some examples of what we would use and what we have in the department. So on the right is a 100-litre bioreactor where we can control things like pH, oxygen, temperature, and stirrer speed. As you can see, it's quite a large piece of kit, so useful when we want a pilot version of a commercial sized vessel, but a lot of work to run and a lot of material would be needed, especially if we're thinking about, we need to screen thousands of different vaccine candidates to make. So going along from right to left, are other vessels that we can use in the department, going down in volume sizes, and also just kind of simplifies the research and speeds it up the smaller scale that we go. So all the way down to microroll plates on the right hand side, where we essentially work at the micro scale and culture in parallel on one plate, for example, 24, 96 or 364 conditions at one time in each of these individual wells. So the ability to mimic behavior at different volumes or scales is a huge advantage in the world of research, as it can really speed up the screening of different vaccine candidates, and it costs much less when you're working at such small volumes. In the department, we miniaturize many different operations. So here are another 2 examples. On the left, we have a miniaturized version of a centrifuge, which is used during product harvest and purification. And on the right is a miniaturized version of a filtration system, again, for us to kind of test different conditions and different filter types, for example. So the main benefit in general of all of these kind of miniaturized systems that we use is to help us predict the outcome of what occurs at larger volumes based on the performance in the smaller volumes. And it's a lot easier for us to work with. So in the context of my research, I have worked on cultivating our pneumococcal conjugate vaccine in small bioreactors up to 200 ml volumes on the left-hand side and down to 2 ml volumes in the microells on the right. So being able to produce our conjugate vaccine from E. coli in both means basically that I can test loads of different things in parallel in super small volumes on the plate on the right. And then I can use this to predict the performance and how it will be in the much more complex and slightly larger volumes in the bioreactors on the left. So in the middle, I've put like a colored representation of our conjugate vaccine that I've produced in the lab, both from the microells and the bioreactors. So the polysaccharide component of the vaccine is shown in green, and the immune boosting protein component of our vaccine is shown in red. So you can see there's 100-fold difference in volume between the microells and the bioreactors, and the amount of vaccine, which we quantify in this image by the band intensity, so how bright that color is and how chunky that band is. You can see they're pretty much the same, which basically shows that I've got a good representation in the microells of my bioreactors between the two scales. So it means that basically I can use these microells to test my thousands of different options and different vaccine candidates, which is fantastic. It's such a powerful tool. So another really powerful tool that we use in our research, and I hope these videos are working for you because it's much more interesting. Excellent. It's much more interesting to see videos than to have me talk about it. We use automation and or robots basically. So I prepared these videos just to show you some of the automated process. The real advantage of these is the efficiency and repeatability of the work that is done on them. So the accuracy of the research is hugely improved when you take out the possibility of any manual error. And again, whilst you're screening thousands of different vaccine candidates in, for example, these miniaturized formats that I showed you before, it's hugely beneficial when the alternative is working manually or 24, 96 or 364 different conditions at the same time. So it really does save my hands a lot of work. And as technology is improving all the time, automation is already and will continue to gain a huge amount of traction. Being able to learn how to use it and program these systems really is such a unique skill, and it's pretty sought after. So during the undergraduate and postgraduate courses, students do research projects, and they can, for example, learn to work with systems such as this on different research projects, as I mentioned, which are designed by postdoctoral students, so PhD students, or with researchers such as myself, for example. So coming back again to my work more specifically, and the combination of miniaturization and automation, the implications in terms of vaccine research, it's just so invaluable when you're having these tools and applying these tools. So in my work, I have used this combination to test different strains of E. coli that make our vaccine. I've tested different nutrients that I grow the E. coli in, and also looked at different combinations of this immune boosting protein component with my pneumococcal polysaccharides. So we are in the early phases of research, but to be part of this journey that is bringing these products to the market and the impact that they could have eventually is invaluable. So the tools and research that we do, I guess, is just a stepping stone in reducing the number of vaccine candidates to test, but our courses are a stepping stone to joining in with our research. So thank you all for your attention. I hope I haven't bored you too much, and hand over to Kim for any questions.
Fantastic, thank you. Thank you very much. That was great. I enjoyed that. What I could watch, because I was also keeping an eye on the questions, which are brilliant. So what I'm going to do is I'm going to invite Jasmine and Martina to scroll through the questions in the Q&A, because some will relate to clarification of some things that you were saying, and others are questions which you might want to group up together. So if I leave you to have a look through the questions, please put your questions. I can see they're coming in thick and fast, which is great. But whilst Martina and Jasmine are looking at those, I just want to remind you, there's another poll that I'm going to launch here. I'd like to know if you are, no, sorry, that's fine. Forgive me. I've noticed that some people are undergraduate and some people are postgraduate. If you're interested in studying MSc Biochemical Engineering with us, Professor Dan Bracewell will be available for a Q&A session coming up. So I'll put a link to that in the chat. If you want to sign up for it, you can ask Dan about the process of doing that. If you're interested in cell and gene therapy, not only have we got a cell and gene therapy taster lecture coming up, but we've also got a Q&A session with Kasim. So you can actually ask him if you want to know more about applying for a program. So I just really want to mention that those are coming up. And finally, I know that a lot of people are interested in studying a degree with us or elsewhere at UCL. There's a UCL Undergraduate Open Day happening on the week of the 14th of June. We're going to be there on the 14th of June. You can come and talk to Dr. Brendan Parker. There's also going to be Chika, there's going to be Fiona. So we're going to talk about the program. You can talk to our careers team. Any questions you've got, if you want to know about the program in more details, we'll be there for that. And I'll put a link in the chat so that you can register for that as well. Jasmine, Martina, if you answer the questions, if you click answering live, then we can sort of move them across and we can try and get through them as we go through. But I'm going to hand over to you. I see we've got a lot of questions. So if we don't get through them all, apologies, we've only got a certain amount of time, but I'll leave it to Jasmine and Martina to go through them. Thank you.
Yeah. Okay, so I was actually reading some of them while Jasmine was talking. I can't see myself in the video, so hopefully you guys can see me. So now I'm going to start from those that were posted first, so just in the order in which they were posted, and what I'm looking at is the really Q&A box. I know there are some in the chat, maybe I can go through some of those later. So there's a question about capacity of vaccine production before and I guess after or currently, right? Capacity of vaccine production. Now this is a nice question, but it's complex to answer without knowing in which country. Obviously, the capacity really changes a lot, but in the spirit of the other questions, maybe I can understand the background. So in the UK, in the world, there are some very big vaccine producer companies with lots of expertise, not necessarily in the same vaccines, which are GSK, Merck, Pfizer. And there are like some medium and smaller size factories as well. So in the capacity of vaccines per country, as I said, really differ, and per continent, I would say, really differs. I think the pandemic has shown, for some of you that have followed the, Europe and, the European situation, for example, has definitely highlighted that it was quite difficult to find, for example, production factories in Europe, right, where AstraZeneca could essentially contract manufacture the AstraZeneca for the AstraZeneca vaccine. So it is really varied. In terms of low and middle income countries, if the question was hinting at that, is Asia. We work with manufacturers mainly in Asia, and they have a very, there's quite good production. The biggest vaccine manufacturers in the world is actually in India, and this is Serum Institute of India. So although probably not very, not well known, right? The numbers, the names I've mentioned before are sort of, big biopharma who with expertise in vaccines. Serum Institute only produce vaccines. So I would say that the capacity really changes. It has changed slightly, meaning that with the pandemic, while before it was really difficult to obtain investments in the vaccine space, now it is obviously, there has been lots of investment and there is lots of interest. So I would say that, now there is a bit, this is the highest, this is higher, the vaccine and the production size are higher in government's agenda. So this is how I would answer the question. So there are vaccines are based on inactivated some RNA, some viral vectors, right? The current COVID-19 vaccines, and the question here is asking whether which approach is easier to mass produce, and which one is best to address variants. So very difficult question. So it's inactivated virus, I would say that there are like more, it's more traditional manufacturing, and there are more issues with the safety. RNA, easier to scale at mass production, but very new technologies. Therefore, let's say that the cost side is higher. Viral vector, there is a few products there on the market, and I would say is a little bit in between, and price is definitely lower than the RNA, for example. In terms of the variants, this is more of an immunology, vaccinology questions, which I'm not sure how, if I am the best place to answer. But what I've heard from colleagues is that in any of the approaches, this is quite difficult, as it would require a change in the original molecule, and therefore then, you know, further studies in process development to support manufacturing. A lot of these are happening in the UK, for example. This is what I know. And work against variants has been, you know, started a few months ago. But yeah, I wouldn't point to one which is easier than the other. Another question is, can you explain the process of all inactivated and live attenuated? How can manufacturers ensure they are safe, such as the virus? Yeah, so this is a question again for more of an immunologist rather than an engineer. If Jasmine would like to read the questions and can provide further details, I would appreciate. This is, I think, the third questions on the QA box. In time, I'll go along with the others. So this is something really, this is another question which I was really keen to answer, which is Pelin, and it says, you know, average time for approval for a vaccine, you know, between development, manufacturing, approval is 15 years. You know, lots of people are worried about how quick was the release of the COVID-19 vaccines, and whether they're linked to obviously side effects in the future, because maybe something has been overseen, etc. So I would mention some important things: the investment and the attention, because of the global health emergency, has been incredible. And so, for example, imagine the full teams of vaccinologists, immunologists, you know, they left whatever they were doing and they focused solely on trying to develop a vaccine against COVID. And the similar happened in groups in different companies. They dropped other types of vaccines they were manufacturing to focus on this. So there was really a very quick reorganization, coupled with significant investments, and this has made a great impact, in terms of the speed at which we could get a molecule, in which we could get the process there, and the regulatory approval, although the regulatory approval is not a full approval, which was what Jasmine was mentioning, in terms of the 15 years. This is an approval for emergency use, which is a little bit different. And in the pandemic, the regulators were in contact with the groups throughout the clinical trials, so they didn't receive data at the very end, right? They had, all the data alongside the process, just to speed this up. Another thing I'm very keen to say is there has been an unprecedented collaboration worldwide between different academic groups with different expertise and industry, brought together by the pandemic. So
just one quick question, which I've noticed, which I think is just like to ask, because somebody's asked whether you'd need to, the question they've actually asked is, would you be expected to understand this if you're a first year? So can you just explain the context of the presentation, Jasmine, if this is, you know, what sort of level it's at, to reassure someone?
Yeah.
So, I mean, I guess as a first year, you have, from what I remember, it's been a while since I did the undergraduate course, but you do have immunology lectures. You do learn about the basics of different vaccine types. You learn about how they infect your body and the reaction they have on your immune system. You might go into a little bit about the different types of vaccines. So when I was talking about the live attenuated and the whole inactivated, and you'll learn what the differences are and what that means. Going into the kind of more nitty gritty of the manufacturing side, it's kind of tied in as you go over the course. You learn about the different unit operations that are used to culture, to purify, and you learn about each of these in more detail as the years goes on. I'm not entirely sure if that's in your first year per se. I think a lot of this talk obviously would mean more the more you go along in your projects. I tried to make it a bit more so that you could understand it without having studied in this area, or at least get an appreciation for some of the kind of aspects that we look at and the problems that we're facing as well, I guess.
I would add that this is the level of this talk is really for a final year BSc students in the year 3 or our level 7, which is the final year of an MNG degree, just because of where things are placed in our degree. That would be able to really sort of understand you know, the broad spectrum of, you know, from the sort of immunology to the process to the issues in manufacturing. Yeah. There are other questions. I don't know, Jasmine, if there is one. There are some that are refers in particular to what you have discussed. So just feel free to interrupt me if you want to answer a question. There is another question from, you know, Colinette that was related to what I was talking before. So the timeline for vaccine manufacturing, and depending on the urgency of what the vaccine prevents or funding, and you know, I think I've answered that in terms of, you know, this being probably both.
Yeah.
There is another interesting question: how fast does it take to retool? I guess change a vaccine production.
Okay.
Vaccine production before. Oh, sorry. I've now lost my question. Okay. How does it take to change a vaccine production facility to switch between different types? And this is really difficult. This is what I was talking about before. If every vaccine needs a bespoke process development, then you'd need you know, some of the tools might be the same, right? But it really depends, right? And I'll give you an example. Some vaccines are bacterial vaccines, so, and are, you know, you need to actually grow a bacterial organisms. In other cases, like the viral vector, most of them are made using mammalian cell host. So you would not be able to grow those two organisms in the same bioreactor if the bioreactor is made of stainless steel. And, you know, when before I was referring to new technologies, responsive technologies, you know, is something that we do lots of research on. There are new bioreactors which are essentially with the bag, which is a single use that is able, where, you know, we are able to use exactly the same infrastructure and just change the bag, and that would greatly facilitate and go around this problem, right? Of, you know, possible cross-contamination and reusing the infrastructure, possibly for multiple products, right? You know, switching quite quickly between one and the other.
I think as well with this question, quite often pharmaceutical companies will kind of stick to their existing processes that they have. So it is more from a money perspective, you know, the time and effort that it takes for them to research new methods of purifying or new ways of cultivation. You know, if something works, they're not going to spend that time and money to change their process. So quite often they'll stick with the same process where they can, whether or not it's for the better or for the worse in terms of product yields. And that's where the kind of Vax Hub, or the hub that I work in and Martina leads, one thing that we wanted to look at was creating these platforms that can be used for different vaccines but still maintain good quality of product and high yields as well, because a higher yield means a cheaper price per dose.
Yeah, I'm going on with the questions here. So there's another question with the emergence of, again, the variant species. What are some of the strategies that we can adopt for development of vaccines so we can keep up with the speed of the virus mutation? Very difficult. And as I say, there is a lot of immunology work that needs to be done. In terms of speed, though, from an engineering perspective, some of the tools that Jasmine has illustrated, like working at a very small scale, working in an automated fashion, allow us to do to screen, for example, right? Lots of different types of strain, right? If this were available from, the cell engineer, at the same time, and optimize it quite quickly. So I would say that some of the tools that we are working on are exactly aimed at speeding up process development. There's another question on edible vaccines. So it's not something that we have studied here, we have worked on. This is where essentially you make some gene modification to a plant, so that I think it produces some sort of subunit structure. And I think, I think that they look really promising. This are my thoughts, and is especially for countries where some of the vaccines really are not stable at at room temperature, and they need fridge, etc. They would make transportation much easier. So I think, and I hope, lots of research is done on that. Is not specifically our, we don't have collaborators, right, working on those, but it's definitely very promising for the future, I would imagine.
Also, to add on to that, Martina, I was talking to one of my colleagues in the department, and they mentioned to me on the field of edible vaccines that they were working with algae, and they'd actually produced a vaccine in algae that was for, I think it was for salmon, and basically the salmon eat this algae which contains a vaccine, and they were able to vaccinate all the salmon. So although not for people, but still this kind of idea of an edible vaccine, and that's something that's going on in our department. So that was really cool. Another example of some of the research going on.
Yeah, that's true. I was forgetting. Yeah, Brenda's research on algae that could go in very well in this in this direction. Let's keep on maybe for a few more minutes. There are some questions, Jasmine, on the raw material used in the pneumonia vaccine manufacturing. And approximately the time. I don't know if you like to answer that.
I actually don't know how long it takes for them to the time that it takes for the Prevnar 13.
Yeah. So maybe I can answer a little bit. So the raw material. So our difficulty in case of the Prevnar is that everything is confidential. So, for example, I have a separate project, a separate Vaccine project with Pfizer on this blockbuster vaccine that they have, resolving some challenges in the process, and even as part of that collaboration, which obviously is with a legal agreement. I know very little about this type of questions, like what are the materials, as well as what is the time. But you think that the starting material for the production of the polysaccharide and the protein is a bacterial fermentation, which is basically within a day. But then following that, there are a number of different steps of purification, which, where, in between those, material can be stored in a fridge if they have tried, and, and that's okay, and it's stable. So that particular process is probably involving thousands of scientists and multi-sites. I don't know exactly, that to start from finish, my guess would be, once you have the two produced, the two polysaccharide and the protein, would be in the order of weeks. But yeah, that's all my guess. So, So we have another question of raw materials used in the pneumonia vaccine manufacture. Oh, sorry, this just, sorry, my thing kept skipping. So how is the vaccine design process like? Do we start with a detailed, well-defined molecular structure and then use different manufacturing to make? Well, it's so there is an initial stage where, you know, there are like experts, like vaccinologists, that they essentially work on the, you know, the initial stage to make the molecule. For example, in this case, like in the viral vaccines and the AstraZeneca Oxford one, they focus more on the actual the genetic manipulation of the virus and do initial small-scale experiments to you know, to ensure, you know, to, for example, to characterize the structure. Then after that, you know, the way in which it is produced is by infecting mammalian cells. So you need to have quite a, you know, a good cell culture number there, and it might take up to 2 weeks. Then the virus is inoculated to infect those cells. All these are sort of the initial steps that need, let's say, some small scale experimentation to optimize it, right? To optimize the media, for example, to optimize the growth condition, to optimize what's the good percentage of a virus versus cells. So all of these will have to be optimized in sort of small scale, sometimes flasks, sometimes small scales bioreactor fermentation. Before, yeah, you then go to the purification steps.
So I'm just aware that we've come to 2 o'clock. So if it's okay, so I feel really bad because we've got so many questions that we'd love to answer. So if it's okay for Martina and Jasmine to stay on, we'll carry on for 5 more minutes and try and answer as many questions as we can. If it's okay with you, Martina, and you, Jasmine, but then we'll finish at 5 past, because otherwise the recording this will be too long and we'll be cut off at the end anyway. Yeah, great. Absolutely.
I'm just going to scroll through questions and maybe talk about something. Yes, find the questions that we haven't touched on, as I think there's quite a few.
In that case, I'll give you a moment to just scroll through the questions. I know that Jasmine and I have both been trying to type answers. Some of them we just can't go into. They're not really to do with manufacturing vaccines. They're great questions, but they're just not ones we can answer. Just a reminder: if you look through the chat, you'll find links to the MSc Open Day, the Undergraduate Open Day. There's a whole series of events that we're doing, and I put a link to a survey. We'd really appreciate your feedback. We'd like to improve these. We've even had a few ideas for future taster lectures, so please do get in touch with us afterwards. You will have my email address after this, so you can always get in touch. But we'll try and get through a couple more of your questions in the next 3 minutes.
Yeah. Right. So right. Yeah. So, some questions I'm not sure, because it just in the way in which they are phrased, I don't think I can provide a very good answer. I'd like to to talk to you essentially. So, for example, if we found a candidate that's been successful in trial, how would you scale up the process? And that a particular challenge that will be faced at this stage. So, so the clinical trials, in order to do clinical trials, they need to have the material needs to be already. So you need to produce the material for the trial, which is usually in relatively large quantities. So this means that some scale up has already been done before, and is already not only has been done at scale before clinical trials, but there is a the material produced for clinical trials in humans needs to be it needs to follow the GMP compliance. So it needs to be according to the sort of certain regulations. There are challenges in scaling up. So, for example, if you have a process that works perfectly at the liter scale, this doesn't mean that when you go to 200 liter, 2000, you have exactly the same product, and looking at, correlations, right, where you can really, have a very good performance at large scale, for first time round is something that in the department we look at, both in the upstream and in the bioreactor space, as I was saying, which is my research, but also in the downstream processing side. Let me find modeling. I can answer the last questions about modeling as different from what we have talked about before. It depends. I think that there are. I don't think that modeling plays a large role in terms of in the first stages, when in terms of the molecule and the immunology. I might be wrong, but this is what Sarah tells me. So I'll go with what she says. Maybe, but then we have modeling in the VACSAB. So for example, looking at how to optimize the supply chain, right? There's been lots of supply chain issues in the current pandemic. So in order to make, once the manufacturing process was ready for a vaccine, then maybe there were not, the certain components needed, some tubes, some bags, some probes to measure pH and oxygen, etc. They were missing just because they were needed in quantities, and the company was not able to produce them so quickly. So there were supply chain issues like that. So some of the modeling that we do is looking at the supply chain, and also looking at how can we best optimize the process based on the data available, so that we don't do different we don't try, right? So essentially to decrease the experimentation load, and try to use such modeling approach. If they're science driven and data driven, they're very effective sometimes. There might be other questions that I'm able to answer. I don't know, Kim, if you want, I can spend a few minutes to type answer this afternoon.
Well, I'll tell you what we'll do. I'm just going to give you a moment. If you pick the 3 questions that you most want to answer, because we have to end it, and when it ends, all the chat will go as well. So we'll pick the two most important questions. But one quick question of Jasmine. Jasmine, do you teach on the undergraduate program? Somebody's asked.
I don't teach, no. I assist mostly with postdoctoral students, so PhD students. I kind of work alongside Martina and help with their training or anything that they need for that. In terms of the teaching, that's Martina's job.
So I can maybe say final thing. Yeah. So in our degrees, they are academic staff and teaching fellow body essentially do the main teaching. So some of the experienced PDRAs and the doctoral students supports us, for example, for projects in the lab. You'll be stuck with us. I think I would end here. Thank you for your attention, and really I'm sorry if
we could only get through so many questions, but they were brilliant questions. They were really, really good. And I have put a link to UCL Mind's coronavirus podcast. You can hear far more about Martina's work about the whole sector. And you've also got Susie Farid as well, Professor Suzanne Farid, who is a professor on our program, and she's got some fascinating things to say. Stephen Morris. We've got a lot of people who've been involved in that, and you can find out a huge amount about what our involvement has been. So do check that out if you have the time. Finally, just thank you for your questions. Thank you for taking part. It's been really great to have you here. And finally, thank you, Jasmine. Thank you, Martina. Have a great afternoon. Very welcome. Thank you for the kind messages in the chat. So have a great afternoon. And we do hope that before too long, we get to see some of you at UCL and get to show you around in person and answer your questions properly. Thank you very much. Bye-bye.
Thank you.
Bye.